Welcome back, AP Biology students. This is Mr. MacKay. Today we're going to talk about non-Mendelian genetics. Yesterday, hopefully you went over Mendel's 4 laws of genetics: law of segregation, law of independent assortment, etc., law of dominance. Today we're going to talk about non-Mendelian genetics. So when Mendel looked at his pea plants, he looked at 7 different traits. And of the 7 traits he looked at, there was 2 possible variations. So for example, stem length, the stem was either short or it was tall. There was nothing else. For seed color, it was yellow or it was green. There's 2 possible phenotypes for whatever trait he happened to look at. In our first example of non-Mendelian genetics, we're looking at incomplete dominance. So you can see now, regular Mendelian genetics, there's a dominant and a recessive trait. There's 2 alleles, which are always shown with an upper and a lowercase letter. So maybe it's a capital A and a lowercase a. Doesn't matter with letters. And there's only 2 phenotypes possible: yellow or green, tall or short, whatever. In non-Mendelian genetics for incomplete dominance, there is no dominant or recessive. They are both of equal strength. We still use 2 alleles, but now they're both shown in uppercase letters. So maybe we have an allele A and allele B, both capitalized. Because of that, we have 3 different possible phenotypes. We could have an AA phenotype, we could have an AB phenotype for whatever these traits represent, or we could have a BB. 3 different possibilities for phenotypes based on this. So I'm going to do one practice question from your booklet page. So this is booklet page 4, which the answer key has been posted. You can do these on your own. So I'll do question number 1. In shorthorn cattle, the white allele, capital W, is codominant to red. So you still have to show your legend. Big W for white, big R for red. The heterozygous condition is called roan, and these cattle have a mixture of red and white hair. What are the genotypic and phenotypic ratios for each of the following? So a roan and a red. Well, it told me the heterozygous for roan is one of each letter. So WR, and a red would be RR. So now we just do our Punnett squared like normal. Notice again, no lowercase letters here. Genotypic ratio then is going to be 2/4 WR and 2/4 RR. Phenotypic ratio is 2/4 roan and 2/4 red. That's it. That's a Punnett square doing an incomplete dominance. Maybe you'll do one more because that was so easy. Let's do the bottom one. 2 roans. 1D was 2 roans. I don't have to repeat the legend. I've already written it out. But I have to show the cross. WR, the 2 alleles. Segregation still happens. The law of segregation, they still separate out. So the W and the R separate from each other in the future gametes, the eggs or the sperm. We fertilize those gametes, meaning we bring them together. And now again, we show the genotypic ratio. This time it's 1/4 WW, 2/4 WR, and 1/4 RR. And we have to show the phenotypic ratio. 1/4 of the offspring are going to be white, 2/4 of the offspring are going to be roan, and 1/4 red. So that's 2 examples of how incomplete or codominance works. Another type of non-Mendelian genetics is called multiple alleles. So you can see at the top, the regular Mendelian genetics, that stays the same. But with multiple alleles, there are dominant and recessive, but now there's more than 2 alleles in the population. Notice both of these other ones, there's only 2 alleles here, capital W, capital R. 2 alleles here, same ones. And in these examples, we could have had capital T and a lowercase t, or a capital G and a lowercase g. 2 alleles only in regular Mendelian genetics. Non-Mendelian genetics, we have more than 2 alleles, although one organism only has 2. Any individual can only have 2 alleles. You get 1 allele from mom and 1 allele from dad. When we show multiple allele symbols, we often do something like this. So an S1, an S2, and S3. That would be an example of 3 different alleles. For blood types, which is your booklet page 5, it's always shown as an IA for allele for blood type A, an I superscript B for blood type B, and a lowercase I for blood type O. Because multiple alleles with blood type also is is codominance. We'll see that in a second. And you get more than 2 phenotypes. So let's do a practice question here using blood types. Let's say dad is homozygous for blood type A. And we're crossing dad with mom, who is heterozygous for blood type B. Normal Punnett square now. The superscripts just stay with the capital I. And this individual, this child, would be IAIB, IAIB, IA little I, and IA little I. So, genotypic ratio. We've got 2/4 are IA, IB, and 2/4 are IA little I. Phenotypic ratio. We've got 2/4 are blood type AB, and 2/4 are blood type A. And this goes back, this AB, these are codominant to each other, like the last example. So blood type is actually an example of codominance and multiple alleles both at the same time. And as you know from the blood unit, you can have 4 different phenotypes: type A, type B, type AB, and type O. And that is the end of the first non-Mendelian genetics lessons. We'll do some more of those tomorrow. Talk to you later. Bye.